Hello, everyone. This is Pocket Sized Bio with Dr. Casco. And today we're going to talk about different variations of Mendelian genetics that we've seen based on monohybrid, dihybrid crosses and how you get one-for-one allele for a particular trait. And I like to call it as variations of Mendelian genetics, where you have different scenarios, like I just mentioned, right? One allele for one trait. And we're going to go through several of these, right? Multiple alleles for one gene, right? Polygenic inheritance, dominance inheritance traits, or variations of dominance. For example, incomplete dominance or codominance, right? And then we're going to get into pedigree charts, right? So let's get to it. This is essentially 5.4 of the Waste Nelson Biology ATAR textbook. All right. So multiple alleles for one gene, right? What is the name essentially tells you what's happening here, right? Sometimes particular traits can have various alleles to for that given trait. And a famous example is the blood group system, right? That we're going to take a look at. So it may contain more than just two alleles for a gene, right? And this is referred to as multiple alleles for a trait, right? So and this is occurring within one individual, right? Um, Generally, there's either or, right? There's two forms, but you could have instances where there's multiple alleles for one given situation. All right. So again, like I was mentioning before, the ABO blood group, right, system in humans, right, and this also shares a characteristic trait of another variant of Mendelian genetics, which is codominance, and I'll get into that later in the video. Right. So in this case, you have a situation where you have three possible alleles for a population, right? And um, what's happening in this case is that you have, the alleles that are being expressed by a particular um, protein, right? Ultimately, a protein is what forms that particular trait, right? So in the case of blood type, as we all know, right, there's several types of blood type. You're right. You have A, blood type B, blood type AB, or blood type O, right? And these particular alleles, they're expressed by having a little marker designated by an I, right? So you have this little I, but this I just represents kind of the presence of the um particular, and it's referred to as an antigen being present, right? And um then the type of antigen that you can have is that regard of A or B that you can see in this figure here, right? Let's say blood type A, you have this particular um a molecule, the shape of a sphere, right? Or a circle there. And then you have one in the shape of a diamond, right? So any individual that has this particular allele would have this letter I, and then it would have is denoted by having this particular allele, right? So this individual will have the A blood type. Now, remember that alleles work in pairs, right? Because of the homologous pairs. So a person that has blood type A would be basically A blood type, right? So I do want to note, though, that a person that doesn't have A or B is referred to as blood type O, right? In this case, you would designate the individual as having lowercase I, right? And it's just a matter that there is no proteins present here, right? So a person that has A blood type will have this particular antigen. A person that has B blood type would have this particular antigen. A person that is AB can actually express both antigens. And this is essentially the idea of codominance, where you could co-express, if I can say, these two particular proteins, right? And then a person that is O blood type has no antigens, okay? No antigens there. And they could be expressed by that lowercase I that you can see here. All right. So back to the idea of multiple alleles. You can be either A, as have that A allele. You can have that B allele, right? Or you can have a combination of both designated as AB, right? Essentially being heterozygous, right? Or you can have an absence or not having any of these alleles, which is essentially that you're not producing any antigen. Okay. So how will this work in a Punnett square, right? It still applies, right? It still conserves this idea of passing down these homologous chromosomes, right, through the gametes into the Punnett square, right? Probability there. So um, uh, let's take a look, right, where you have this situation. And let's say you have an individual that would carry or that has AB blood type, right, and is crossed with another individual that is AI blood type. So in this case, you can have an individual that is, as the little diagram shows here, your capital I and your B, and then you have a little I here, right? So the individual would be, right, capital I with that little b. So this particular individual will just have a B blood type, right? And this individual that you would see here, A and then B here, would be a B blood type. Remember, always following the rules of the Punnett square, you would put each allele, right, through independent or through segregation. And let me just put that line here. And then you just make a cross, right? And you will plot out the Punnett square. In this particular quadrant, you'll have a probability of getting a B blood type. Okay. You can have in this quadrant having BB blood type, which would just be will just become B blood type, right? And so forth and so forth. If you were to have an I, right, you would be A blood type because A just shows that you have the presence of an A antigen, right? So, in other words, you basically are AO, but since any presence of an antigen, you take that blood type. So in this case, it would be A, right? Or in this case, it would be B. So what would happen? How does it come out in this case, right? In terms of genotypic and phenotypic ratios, you would basically have a 1 to, in terms of genotype, right? 1 or 25% of AB, 25% of BB, right? 25% of AI and BI and so forth. But ultimately, in terms of phenotypic ratios, you would be, you will have one blood group AB, two blood groups being B, and one blood group being A. So if I were to highlight this, right, this individual and this individual are pretty much identical. They're the same thing, right? So in the population, there are 4 possible subtypes, right? And there's no intermediate, okay? And we're going to get into intermediate forms of certain traits later on, right? And this type of inheritance pattern, right, is or having many alleles, is referred to as being discontinuous variation in the population, right? And what it means is discontinuous is that there's no gradient, right? There's no partial A and partial B situation going on, or you have a sudden blend throughout the population. where you're going to be, let's say, like full A or mildly A and then turning into mildly B, it's either or, you know, alleles. So in this case, when you have this type of situation, right, um, you refer to this as being a discontinuous variation. Okay. And because it's being controlled by a particular allele. So in the population, you would plot this type of frequency as a bar graph, right? Either you're a discrete, phenotype, in this case, B, AB, or O, right? In a certain percentage in a population or whatever type of quantification you're doing, right? Um, it would show this discontinuous variation. Okay, so this refers to as multiple alleles, right? You have several forms for one particular trait. Okay, we're gonna get into another um, characteristic, right, of inheritance, which is polygenic inheritance, where you have many genes, right, um, for a particular trait, right? Many different genes for a particular trait. And here you have multiple alleles for one gene, right? Multiple forms of one particular gene. And here you have many genes for one particular trait, right? And this is called polygenic inheritance, right? And the name tells you, you know what it is. So you have more than one gene affecting the outcome of a particular characteristic. Sorry, I've been mentioning traits. It's actually characteristic. For example, skin color, right? Skin tone, right? So many genes equals one trait or characteristic. Okay. Um, So polygenes, you will see this often, right? Or you have polygenes. It's just refers to genes that add a small effect to a characteristic, but when you have many of them, they start to give you that overall feature, right? Um, and several examples of this is human height, right? Skin color would be another example. And in this case, you would see some behavior or some within the population, you'll see a gradient of the trait, right? It's also referred to as continuous variation, right? And generally, when you have a characteristic that have more genes, because there's more combinations of genes that are that could be dominant and recessive, right? Um, you'll tend to have more gradient because there's just more combinations, right? Uh, so the more genes you have, the more of a gradient there is, right? And of course, in some with some traits, you do have an environment contributing to the particular trait, right? Nutrition, right? Exposure to sun, for example, and things like that. In the case of height, there's approximately 200 genes that contribute to that magnitude, right? If you were to take a look at the picture here, um, now you don't need to worry about this type of Punnett square, right? This is while it is possible for you to do, you definitely won't be doing these type of crosses, right? But what I want you to understand is that here you have three different genes designated by A, B, and C, right? Where you're seeing different alleles, dominant and recessive alleles, and all these combinations of a dominant form and um, a recessive form when you're crossing together and you're getting that homologous pair, right, of the offspring, you could see that in the population, you're going to have a particular gradient, right? So that's why it refers to as being continuous, a continuous trait of variation, right? Because you're seeing more variations of that trait within the population, right? And depending on the recessiveness and the dominance that is expressed in the population, generally you'll get a bell curve, right? Um, showing a percentage. And generally also these types of continuous variations will be plotted as a histogram, not as a bar graph. Okay. So keep that in mind there. The book has a very nice table that you can distinguish between continuous variation and discontinuous variation, right? Type of inheritance, in this case for continuous, would be polygenic inheritance. Discontinuous variations would be single gene inheritance, in the case of multiple alleles, right? Causes, of course, both of them will be influenced by genetic factors, but continuous variation will tend to have the environment kind of influencing some to some degree, right? And what's happening, more specifically, right? You'll see gradual changes within the population, right? From one trait to another, differences are slight along a continuum because there's so many genes. You have a gradient, a nice gradient blend, right? Um, in the case of multiple alleles, it's either or, right? It's clear, concrete between the traits, right? There's no intermediate form. As I mentioned before, you have a histogram versus a bar graph, right? Between polygenic inheritance and a multiple allele situation, right? And of course, some examples. It's good that you learn some examples, right? To discuss height, weight, skin color for gradients, right? And for example, the ability to tongue roll, having something present or not present, attached or detached earlobes, blood groups. It's all a fixed, discrete thing, right? So this is polygenic inheritance, and um, be sure to be able to distinguish between continuous and discontinuous variation there. Okay. Getting into more specifically dominance, right? Inheritance patterns. So again, as I mentioned, there's some kind of variations in Mendelian genetics, and um, and this moreover comes into play with dominance, right? And there's incomplete dominance and codominance, right? That we're going to take a look um, at each one. So in the case of incomplete dominance, the name kind of tells you what's going on there. That um, you really don't have a dominant allele, right? There's no dominant or recessive one at that, right? Each allele contributes partially to the particular trait, right? So, again, similar to normal Mendelian genetics, each letter represents a characteristic trait. Okay. Um, and for example, in this case, you have this situation with the snapdragons where you can have certain variants of the snapdragon flower where you have some white flowers and then you have some red flowers and then you have an intermediate of them, right? An intermediate form which is pink, right? And here in this case, you have this little notation there, but that's not as important, I believe, where you would just have a characteristic C there that either you're red or white. You can't really call it dominant because they're not dominant, right? You can't say it's a capital C and lowercase c because there is really no dominance, right? So that's what I'm trying to mention here, right? So either you're red or white, but if you're heterozygous, that you have an R and a W, you end up being pink, right? So let's say you were to cross a pink with a pink, you would get this nice little gradient here where you would have 25% of the offspring being red, basically big R here, sorry, red as the designated letter. You'll have 50% of them being pink because you have a white and a red alleles. And then you have white, which you have WW being 1/4, right? So yeah, this refers to as incomplete dominance where you have an intermediate blend between the two um uh characters traits before the two traits, right? Between the two traits. So, and then you also have codominance, right? Codominance is where both alleles are fully expressed, right? And observed in the phenotype. So there's absolutely no dominance. They're both essentially dominant, right? And they're both expressing their protein, if I can say, right? And again, like I was mentioning, blood type is a perfect example of codominance, where you would have the A antigen protein and the B antigen protein both fully expressed on the surface of the cell, and there's really no a sense of dominance there. Other examples could include the coat color, right, of cows, where you have a white bull that is W and W, right? Again, same situation here, where that capital C is just kind of like a holder for the letter. For the actual allele, not as important again. And then you have a red cow, right? Uh, um, big R, and you breed them together, and then you'll have a particular outcome where you'll have a brownish coloration of spots and a white coloration throughout the skin, throughout the overall skin there. Now, what patterning is a different situation there, but um, different set of genes being implemented there. But what's important is to show that both um, colors are being expressed, right? So you have white and white. Basically, you would have this black and white coloration, and then you have a heterozygous individual where you have, black and brown, right? And then you have red cattle, which you have a that dark brownish appearance there. All right. The maroon appearance there. Okay, so codominance, right? Codominance. Both alleles are fully expressed. Okay. So the book has nice summaries of tables for you guys to take a look at. Um, I strongly recommend you guys study these, and then we can take a look at it in the classroom, right? Complete dominance, incomplete dominance, codominance. So complete dominance refers to the Mendelian normal one, right? That you'll think about. Um, and then you have particular examples, right, of how to annotate, right, and how the letters will look like whenever you're studying these particular situations. Okay.